welcome to follow us on Twitter, and please feel free to contact us with any questions or suggestions at our email address. All the links will be posted in the chat shortly. If you have any questions during the talk, please submit them in the Ask a Question section at the bottom of the screen, and we'll address them at the end. Finally, we'll be hosting a more intimate discussion on Zoom with Professor Vogt a few minutes after the end of the seminar. Feel free to join. The link will be in the chat. And now it's my absolute pleasure to introduce Katrin Vogt, who received her Doctor Rerum Naturalium at the Max Planck Institute of Neurobiology in Germany, where she investigated how multimodal sensory information is processed in the brain and associated with reward or punishment under the guidance of Professor Hubner. She then did research as a visiting scientist at Genelia in the lab of Gerald Rubin. This was followed by a postdoc in Tohoku University in Japan with Professor Tanimoto and a postdoc in Harvard with Professor Samuel. In 2020, amidst the challenges of COVID, she opened her own lab at the Department of Biology and Center of Advanced Study of Collective Behavior in the University of Konstanz in Germany. Her group is interested in deciphering the neural mechanisms that underlie behavioral flexibility towards different external cues and in different internal states. In order to perform this research, they take advantage of the versatility of Drosophila larvae and how they lend themselves to various genetic and imaging techniques. In any case, this is no small task, and I look forward to hearing all about it. Please, Katrin, the stage is yours.
Okay, thanks a lot. I hope you can hear me. Yes? Okay. Good. Yeah, thanks a lot for the invitation. I'm really excited to talk here today. And first, I will talk a little bit about my postdoc project that I did in the US in the Samuel Lab, which has just been published last year. So it's still kind of new. And also, it's the basis for the work we're doing now in my lab in the University of Konstanz and the Cluster for Collective Behavior. So yeah, first a little bit of the background, the work I did before, and then I have a little bit of new data for you from my own lab. Okay, so I have to click here. Yes. So yeah, I'm interested in decision making and how flexible is this decision making, and how can we be such flexible? And what I really like, what is a really nice example of flexible decision making, I think, is foraging in humans. So if you go to the supermarket, I think if you think about it, you never buy exactly the same things in the supermarket because there's always something different. And there's a lot of things that influence your decision making in the supermarket. For example, the context can change. So if something is on sale, you might buy more of it than you eventually wanted. So it will change your decisions. During the pandemic, people made a lot of different decisions by just hoarding stuff and buying all the toilet paper. Again, a change in context that affects your decisions. Also, your experience can affect which items you choose in the supermarket. So if you bought something and you didn't really like it and made you sick or something, you will probably not buy it again. Again, a change in your decision making. And then I think also something that really everybody knows is that our internal state definitely affects our foraging behavior. So if we are hungry and we're going to the supermarket, things will look very different. And what happens here is that even though we go to the same supermarket with the same sensory cues, the same items, something happens in our body and in our brain that affects our behavioral decision. So when we're hungry, instead of buying all the vegetables and the healthy stuff, we will go for like the chocolate and everything and just buy much more things. So clearly our internal state, hunger, really affects our decision making. And so now I'm interested in what's going on in the brain here. How does the brain process this information, and what's changing in the information processing in a change in internal state? And this is really hard to do in humans because, it's not easy to manipulate neurons or the neural system in humans. So I chose to work with the Drosophila, with the fruit fly, which still we share a lot of genes with humans, but the brain is highly reduced in numbers. So the adult fly has about 100,000 neurons, and my choice of model organism, the fly larva, we go down to 10,000 neurons. So it's reduced in numbers, but the brain still is quite complex and it's still quite interesting. And we were wondering, can the fly larva with a brain of 10,000 neurons, does it also have this influence of internal state on its behavior? So if we send the larva to the supermarket in a hungry state, would they also go and buy all the chocolate? So of course we don't send them to the supermarket, but we can ask them if they like, if their preference for a stimulus changes, like for example for an odorant. And that's what I did at the beginning of my postdoc. So I took a group of larvae, put them in this Petri dish. So this time I took them just from the food, put them on a Petri dish and added an odor and see if they approach or avoid this odor. And I think what you can see here is that they really didn't like this odor, which is general acetate. It's a green leaf odorant. It's in green leaves and lemongrass, things like that. So it's not really food for them. Kind of makes sense that they're not really excited about it. However, if I take larvae that I have food deprived for a few hours in water and repeat the exactly same test, so it's exactly the same odorant, it's the same Petri dish, but this time the larva is hungry, and then we see that they are attracted to this odor. They really go and check it out, which I think kind of makes sense, that they expand their food of their choices, and they have like more things to choose from and more possibilities to find food also in nature if they overcome these avoidances. And we still don't know if they can actually feed on green leaves. That's a question that I often get, but we will probably look at this at some point, but we didn't do that yet. It would be interesting if they could actually, if this would actually be nutritious for them. So, but yeah, I think we all agree that also the larvae can change their internal state. They can become hungry, and then they also change their behavioral decisions. And I think if you would put them to the supermarket, then they would go more for the chocolate too. So that works. And so now, since this is a nice model organism and we have a lot of tools, we can now go and really look also what's going on in the brain in this case. And just a little bit background for Drosophila. So in Drosophila, we have a rich range of genetic tools that we can use to manipulate and visualize single cell and cell components in the brain. It's the GAL4-UAS system. It's basically like a mix and match card game. So one system is the head of the animal, one is the tail of the animal, and you can combine them in each combination you want and come up with basically different types of flies that you can create. So for example, the head would be the driver line, the GAL4, where we can choose where we want to express something in the brain, like the olfactory neurons, the visual neurons, mushroom body, the learning center. And the second part would be the effector line, the UAS, where we define what we want to do in these neurons. So we can visualize them with GFP, we can silence them with, for example, tetanus toxin or other effectors. We can also activate them with optogenetics or record activity with GCamp. And so we can combine these driver lines with our effectors and really look at all the different characteristics of a cell type, for example. And another thing we have in the larva, and by now also in the fly, is that we have a complete map of all the neural circuits in the brain, which of course makes it also easy to dissect the neural circuits and to understand the dynamics of the neural circuits. And this is all thanks to Albert Cardona, who was back then in Janelia Research Campus, who initiated this whole connectome initiative. And I was lucky when I started my postdoc that the Samuel and the Louise lab, that they started to look at the olfactory system, and they traced all the neurons in the olfactory system. So on the right, you see the head of the larva with the dorsal organ, that's the nose of the larva, where the olfactory receptors sit. And then the antennal nerve brings the olfactory information into the brain. And there it goes to the antennal lobe, the AL here. And you see there's a lot of cell types. So there is some processing going on already in the antennal lobe. And also the neurons, projection neurons, bring the information to the higher brain. And this is the green and the blue ones you see that go to the mushroom body calyx and the lateral horn. So this is basically the olfactory system. Here it's a bit in a reduced version. So you have the olfactory receptor neurons, information goes to the antennal lobe. There is some processing going on, but then information is going to the higher brain via the projection neurons to the lateral horn and mushroom body calyx. And then the idea is that the order preference is happening in the end, like at the output of the lateral horn and the mushroom body calyx. Okay, so this, and what is also nice, and which makes the Drosophila and the Drosophila larva even more valuable as a model organism, is that, for example, this architecture of the olfactory system is very similar to mammals. So in mammals, you also have olfactory receptor neurons that go to the olfactory bulb, where you also form these glomeruli structures that we have also in the larva. And then again, there's also projection neurons that bring the information to the higher brain, like the piriform cortex or the amygdala. So it's a very similar architecture. Okay, and now what I wanted to know is, so where in this circuit, where in this system does the internal state information come in and modify the processing of the olfactory information? Is it happening at the receptor neuron level? Is it in the antennal lobe? Is it happening in the higher brain? And to address this question, I performed functional imaging in the fly larva, which is really easy because they're transparent, so you don't have to cut them open. You can really just use the whole intact animal. And what we did is we used a microfluidics chip. So, and it looked a bit like this. So we have syringes with odor solutions. They flow into this chip. And in the chip, we have different channels to see different odorants. And then they flow by this channel where the little larva sits. And we can expose the larva to the different stimuli. This is what it looks in real. But this is the system that I used in the US, and we are right now also setting this up in Konstanz. So hopefully next year, some point, we can also start imaging here. And yeah, so on the left, you see the syringes and then tubes that are going into the microscope. And in the middle, you see the chip sitting on the objective. And on the right, you see how it looks like if a larva is inserted into the chip. And from the right, we would have the odor channels that, yeah, we can present the odorants to the larva. And so what I did now is I looked at the odor responses in fed and food deprived animals to see if I see anywhere in this olfactory circuit a difference in activation in the different states. And so this is kind of what it would look like. So here we have a driver line for the olfactory neurons, and I just visualized them here with a GFP, or yeah. So in pink, you see all the olfactory neurons. One of them is highlighted in green. You see the dorsal organ at the tip, at the nose of the larva, the dendrites, the cell bodies, and then the axon projecting to the brain. At the bottom, you see the antennal lobe with the different glomeruli structures. And so I imaged in the antennal lobe to see if the output of these neurons is already modulated in a different state. And it's not. So it's pretty much the same, the peak response. And also we looked at a few other, so this is one olfactory receptor neuron, and we also looked at other olfactory receptor neurons that get activated by geranyl acetate, and we usually didn't see a big difference. So it seems like that the output of the olfactory receptor neuron is not affected by internal state. And then the next step is, so this was the input to the antennal lobe. The next step was to look at the output of the antennal lobe, so the so-called projection neurons. And here we looked at the first uniglomerular projection neurons, which sample from one olfactory information from one olfactory receptor neuron in the larva and project this to the higher brain. And there's 21 of them for the 21 olfactory receptor neurons. And surprisingly, what we saw is that there is an increase in response to odorants in the food-deprived state in these neurons. And yeah, they respond more. So we concluded that somewhere in the antennal lobe, internal state is integrated into the system and affects the olfactory processing so that we get this different output in the food-deprived state. And so we can look at these neurons. This is how they look in the EM reconstruction. There's highlighted 4 of them, the blue ones, which respond to geranyl acetate. And we can also look at them in the brain. So this is a GAL4 line labeling the uniglomerular projection neurons. You can also see the cell body, the overlap with the antennal lobe in magenta with the orange, and then projections to the higher brain. And then what I also did is I manipulated these neurons and silenced them. And what we could see here, that indeed these neurons that are more active in the food-deprived state, they are also required for the food-deprived behavior. So the attraction to the odor is suddenly gone when we silence these neurons. And then what was surprising is that though not all the behavior is gone, so we still see the odor avoidance in the fed state. And this was a bit surprising because we thought that all the olfactory behavior response would be mediated by these neurons, but they could still do something. So we were looking for other neurons that could be necessary for this avoidance in the fed state. And yeah, and then thanks to the connectome, basically, we knew that there is also multiglomerular projection neurons that sample from several organs and that project to somewhere else in the brain. So for example, we found one of them I'm talking about today, the cobra MPN, which projects to the mushroom body vertical lobe, so the axons of the mushroom body. And then we also looked at the response of this neuron to the odor in the fed and food deprived state. And here we saw that the activity of this neuron is decreased. So it seems to receive inhibition in the food deprived state, which is the opposite that we saw before. And then we were also interested. So here you see it again in the EM data. You see it's going some other place than the gray neurons, which would be the uniglomerular projection neurons. And also we can visualize this with GFP. You see the neuron here, it overlaps with the antennal lobe. And we also silenced this neuron. And indeed, what we saw that suddenly now the larvae were always attracted to the odor, even in the fed state. So these neurons clearly do the other job they are required for the odor avoidance. And so if we sum this together, what's happening is that in the fed state, we have activation of both outputs. The uniglomerular projection neurons rather weak, and we also see that the multiglomerular projection neuron output is a little bit activated. And in summary, we get avoidance to the odor. However, in the food deprived state, we see a total change in these activity levels. So the UPNs are boosted and are more active, and the MPN activity is decreased, so it's less active. And then generally, we see the switch toward attraction in the behavior of the animal. And so this was super cool, but also we wanted to know, so what's now actually going on in the antennal lobe? What's causing all these changes in activity levels? So we first looked at the uniglomerular projection neurons, which respond more in the food deprived state. And then there is a lot of local inhibitory neurons in the system, I think similar to the olfactory bulb, but we didn't really know much about excitatory neurons that could boost the behavior. So one idea was that maybe it's a neuromodulator like serotonin, which also has excitatory receptors. And so we looked for that. And again, this is another data set where you can see that in the food deprived state, we see this stronger response to the odor. And so what we did now is we manipulated just the serotonin receptor in these neurons. So we used the CRISPR-Cas9 system to knock this receptor out to see if this is required for this boost in activity. And this was in collaboration with the Rosberg lab from Brandeis. They made the CRISPR-Cas9 line. And again, indeed, what you see is that without the serotonin receptor, it's the 5-HT7, it's an excitatory one, we do not see the increase. So it's clearly the serotonin that is required for this boost in activity. Okay, so we have one solution for the UPNs, it's the serotonin. But still, we were wondering, so what's happening with the multiglomerular projection neuron? What could lead to this inhibition? And since this is not a global inhibition of the whole antennal lobe, so there is global local inhibitory neurons that are GABAergic, but since this is not a global inhibition, we thought that those might not be the ones that do this inhibition. But we also know from a paper from Rachel Wilson a while ago that there's also glutamatergic inhibitory channels that are activated by glutamate, so chloride channels that are activated by glutamate that can also perform inhibition on these neurons in the antennal lobe. And so we used, again, we usually see this decrease, so a reduction in response in the food deprived state. And what we did now is we used RNAi to knock down the specific glutamate receptor. It's the glutamate-gated chloride channel, gluCl alpha. And again, what we see is that now if we take out this inhibitory glutamate receptor, we do see that the neuron responds to the odor in both states. So again, this inhibition is mediated via glutamate. And we already have some idea about where this glutamate comes from, because there is so-called picky local neurons, which are glutamatergic and release glutamate into the antennal lobe, and also do have synapses with this multiglomerular projection neuron. That's all something we know because of the connectome. So this totally makes sense. But yeah, so where does the serotonin come from? And then there was also one specific candidate that we thought, okay, this is the one that must be it. So it's called the CSD neuron. It's the contralaterally projecting serotonergic deuterocerebral neuron. It's a very long name. It's in a lot of insects. So it's conserved across insect species, and it's very strongly kind of affiliated to the olfactory system. So you can see the cell bodies next to the antennal lobe, and then it projects up into the higher brain around lateral horn and mushroom body region, goes to the contralateral side of the higher brain, and goes down to the contralateral antennal lobe where it has most of its outputs. So it's very much connected to the olfactory system. And we also do have a driver line for this neuron, as you see in the middle. And so if we now manipulate this neuron, we wanted to see if this plays a role in the food-deprived enhancement. And so first of all, we looked at the activity of this neuron also, and it has an increased odor response in the food-deprived state. So it fits that it will also boost then the uniglomerular projection neurons, because it seems to have also an increased release of serotonin. So that kind of fits. And then we also manipulated this neuron, we silenced it, and we see that this neuron is strongly required for the food-deprived switch to attraction behavior. So this seems to be really, really the key player. So we can now add this to our scheme. And so we see that the CSD is, as a serotonergic neuron, probably releases more serotonin in the food-deprived state, activates the UPNs more, and thereby switching towards increasing the attraction pathway. But we were wondering if the serotonin from the CSD neuron maybe also affects other neurons in the antennal lobe, like the picky local neurons and the MPN, for example, because we do see synapses in the connectome here. And another funny thing is that there actually are no synapses to the UPNs in the connectome, but we think that's happening via spillover in this highly excited state when they're food deprived. Okay, so but yeah, is CSD actually the key switch? Is CSD the neuron that elicits all the changes we see in the antennal lobe? And to test this, we went and optogenetically activated this neuron in fat larvae. So again, normally the fat larvae, they avoid the odor, as you see here, and the food-deprived larvae, they are attracted to the odor. And now if we take fat larvae and activate the CSD neuron, so this is what we saw, that indeed larvae now are actually attracted to the odor. So the activation of the CSD neuron is sufficient to induce this food-deprived behavior, which is pretty cool, I think. And yeah, so what we think is happening is that the CSD, which has actually synapses with these picky local neurons, in the end manages to disinhibit some of them so they can eventually inhibit the MPN in the food-deprived state. And it also, because it's highly active, there is some spillover or extrasynaptic release of serotonin that then also can affect the UPNs, where we didn't find synapses in the connectome. And thereby, it's basically totally reconfiguring the information processing in the antennal lobe. And so there's two take-home messages I have from this study. So first of all, the connectome is awesome, but because it reveals us new cell types, like the multiglomerular projection neuron and the picky local neurons, like we learned a lot about the diversity of cell types that we actually have in the larval antennal lobe. And of course, we get to know all the synaptic connections, which is great. and also very complex, also in the larva. But we should not forget that especially neuromodulation can happen non-synaptically. Like as we showed, that there's probably happening some spillover and extrasynaptic release. And I think it's also a very efficient mechanism because you can change the processing in a circuit without having to grow new synapses or, without structural changes. So it's a very, very plastic and flexible adaptation. And the second thing is that what was also kind of interesting is that the serotonin released from this neuron, it's a single neurotransmitter, but it can have different effects on the downstream neurons. So the synaptic connection to the picky local neurons, it happens through an inhibitory serotonin receptor, the 5-HT1A, but the excitation, the boosting of the UPNs happen through an excitatory receptor. So a second take-home message is, even though sometimes the neurotransmitter of a neuron, you don't really know what it's doing to the downstream neuron, because there can be different receptor types. And so you really need to know all the information. So in addition to the synaptic connections, to reveal these non-synaptic connections, we really need to know the receptors that are in the vicinity of neuromodulatory neurons and which receptors these are. So they can be inhibitory or excitatory. So this is take-home message from the study. And so you might think, okay, this was a lot of neurons. It's a quite complex circuit, even though it's a small larva. And so what we did in collaboration with the Pelevan lab in Harvard, we took all this information from our data, which is a lot of information, and put it in this very constrained model, computational model, to really see if we can reproduce the dynamics of the system. And so we use the actual synaptic numbers for the synaptic weights. We knew the sign of the connectivities based on our studies with the receptors and to see which neurotransmitters the neurons have. We included a non-synaptic connection, and we kind of included the activity levels of the neurons. And so if you run the model, you can actually reproduce all the effects we see. And what was really nice to have all this information in a model. So for example, we could manipulate neurons that we don't have a tool right now, a genetic tool to manipulate them. And we can make predictions about what would happen if we actually do this in the larva. And this will also be nice in the future. Hopefully at some point we get these lines that we can then really also verify this model and verify these predictions. So this will be interesting to see. Okay, so that's so much about what is already published. So we used, so I think the Drosophila larva is really cool. So we can get the neural activity of neurons all over the brain with a very simple and non-invasive setup. We have the connectome, so we can get all the neural connectivity of the whole brain, basically. The behavioral output is easily trackable, I would say. It's very simple experiments, but we get very flexible behaviors. We can use the rich genetic toolkit. We can check for neural requirement, neural sufficiency. And in the end, it's really helpful to be able to use neural network dynamics, like building this dynamical systems model and really verify all the results you find. And so if you want to read more about this is all published last year. That's all the work from my postdoc. And yeah, feel free to check it out. And now maybe to the more interesting part, at least for me. So what about the current projects that we have in the lab? And so I will today talk a little bit about the open questions we still had from my postdoc paper. And so this is mostly work done by my first bachelor student, Nora, that you see here, who did her bachelor thesis last year in my lab and is still around as a research assistant and still producing a lot of great data. So I just showed you that by food deprivation, we can switch the larval behavior from olfactory avoidance to olfactory attraction, and then serotonin is basically the key player here. But we didn't really, or I didn't really have looked into like what kind of is it like all kind of food deprivation? Is it like does it necessary to food deprive the animal of all food, or can we like have some food components provided during the deprivation period, and will they still show the behavior? So we first were a little bit interested in, like, is this specific to full food deprivation, let's say like this. So what Nora did is she ran basically now our control groups, so where we have larvae always on the normal fly food. Then she had the control where the larvae are totally off food, so just in pure water for 5 to 7 hours. That's our usual starvation time. And then she also had a group where she put the larvae in fructose water for 5 to 7 hours, so they had full access to carbohydrates but no proteins. And then we also had a group where we exposed the larvae to protein only. So yeah, no carbohydrates, but we used some rice protein powder that had 92% protein. So I think that's pretty clean. And so she performed all these deprivation with food components and full food deprivation, and then did the usual journal acetate preference test to see if the larvae are attracted or avoid the odor in the end. So are they in a fed or in a food deprived state, basically. And so this is the results. And it was kind of interesting to see that it seems like both fructose and protein are somehow satiating for the larva. However, only protein really significantly, only the larvae that were fed with protein still have a significant avoidance to the odor. And the fructose is like a little bit in between. So it seems like really protein is really important. And fructose can substitute a little bit for the effect, but not fully. So I think it's good that I started off with the full food deprivation, because only there we get the full switch in behavior. So that's interesting. And another experiment, another thing we were interested in is how can we reverse the behavior back to the avoidance of the larvae? So what do we have to feed them? How long do we have to feed them to bring them back to the avoidance behaviors? Like, how flexible is the system on which time scale? And so what Nora did here is, again, we have our two control groups where the larvae are all the time on food or all the time on plain water. And then we had one group that we also had the larvae on water for most of the time, for like 5 hours, 7 hours. But in the last 10 minutes before the odor preference test, we put them on the fructose or on the protein to see if this short refeeding, so they should be starved, they have been in water for many hours, and if this short refeeding is sufficient to bring them back to the fed behavior. And this was really interesting. So what we saw is that protein on this short time scale really has no effect on the behavior. They were totally attracted to the odor, similar to the fully food deprived control. But feeding fructose for 10 minutes was sufficient to fully reverse the behavior. So they were behaved exactly like the fed animals and reversed the behavior. So this was pretty cool. And I think it makes sense that fructose is more quickly accessible to the animal, and thereby it can also have this quick effect. But it was a bit surprising that it totally reverses the whole thing. And then we were wondering, is it actually the sweet taste of the fructose, or is it really the nutritious value of the sugar that elicits this effect? And therefore we tested sorbitol, which is like a sweetener. So it's sweet to the animal, but doesn't have a lot of nutritious value. And indeed, this is not sufficient. So the sweetness alone is not sufficient. Somehow the larva has to detect or quickly detects the nutritious value of this sugar. So this was pretty cool. And in the larva, there is, so far as we know, only a single receptor for fructose. So we went ahead and tested if this receptor is actually also required for the effect. So again, here you see the control. The larva here can sense fructose, and they reverse the behavior. That's a new data set, so we could reproduce that, which is cool. And then in this mutant, for this GR43A fructose receptor, we saw that they at all didn't reverse their behavior. So they really showed attraction to the odor, which is quite interesting. And opens like a whole new field of study for us to really check where exactly in the body, in the brain of the animal, is this receptor required to sense the fructose. There is receptors in the brain, there's receptors in the mouth, in the pharynx for the fructose. It's always the GR43A, but we have the tools to manipulate them independently. So in the future, we will really look like where is this happening? Where does the larva sense the fructose? And then, of course, also we would like to know how is this integrated, or how does it modulate the olfactory processing? Because what we see is a switch in olfactory preference behavior, and how is this all coming together? And then if you look at the olfactory system again, we know based on the connectome that already in the antennal lobe, there is gustatory sensory input that could be fructose input already, so it could directly affect the antennal lobe. We know that the CSD receives non-olfactory sensory input, which could be gustatory, which could be fructose, so it could directly affect the activity of the CSD neuron. But then also in the higher brain and around the mushroom body and the lateral horn, there is also the gustatory information reaches there and could also affect the behavior at this level. So this will be interesting to see in the future. Then another thing is that we're right now interested in is to see like all the pleiotropic effects of food deprivation. What I showed you so far, it affects olfactory behavior. Yes, but it actually affects also all kind of other behaviors in the larva. For example, locomotion. So there is a little bit of data already in the postdoc paper that I showed you, but we looked at this again in a bit more quantitative way. And so how the larva normally walks is they run, they move straight, and then at some point they stop, they wiggle their head, sample, and then often initiate a turn. And then they run again, and then they stop, sample, turn, and then they run again. So it's this very generic pattern that they usually have: run, turn, run, turn. And what we can measure is, for example, the run speed. We can measure the turn angle. So how strong do they turn? And yeah, so this is mostly what we do right now. And we use this new tracking system from the cousin lab here in Constance. It's called Trex. It was made for fish, so somewhere here you see that says fish, but it works perfectly also for fly larvae. And what we get is really, we get the XY positions of the animal, we can get the speed, we can get, as you see on the right here, the head bending angle of the larva. So it gives us a lot of information, and we can really quantitatively analyze this behavior. And for example, what it also gives us is the size of the animal. So we can control for that now, because also we see that the size really affects also the speed of the larva. So the bigger they are, the faster they run. So this is really an important control. Sorry. Okay. And then just to show you that now we also have bigger arenas with 25 centimeter and 25 centimeter to get more of the actual locomotion behavior. But still in this arena, we can reproduce the olfactory preference switch. So from fed to food deprived, as you can see here, which is nice. So we can still use that arena for that behavior. And then we looked at these different locomotion parameters. So if you look at the speed over hours of food deprivation, we see usually that after a short food deprivation of 5 hours, what I just showed you, what we usually do for the olfactory preference behavior, we see an increase in speed. But if we starve them for longer, and we can starve them even longer, but for 20 hours, you can see that they can go back to a decrease in speed again, which we think is just because they're losing energy and they cannot maybe run that fast anymore. Or they start saving energy because it's a really long time for them being food deprived. And what we also see is that the neighbor distance increases with the amount of food deprivation. So they disperse more, they spread out more, they sample more, which I think also makes sense that the more food deprived they are, the more motivated to explore they are. And the head cast angle actually decreases, which kind of fits with the increase in speed, for example, because we usually see if they increase their speed, they also stop less and turn less. So that fits together. So, and this is something that's very reproducible with starvation. So we always see that short-term increase in speed, the decrease in head cast angle, the increased neighbor distance, and we still don't know like how the locomotion circuit in like neural wise is how it's modulated here. So this is something we want to look at. Serotonin might also play a role here, but I don't think it's the CSD neuron. Actually, if we activate the CSD neuron with in the optogenetics experiment, we can induce the food deprived olfactory behavior, but we don't see this in increase in speed, for example. So the CSD seems to be really just for the olfactory behavior. So maybe there's another serotonergic neuron that is modulating the locomotion system. Okay, and then a last point that I would like to talk about today is that I mentioned to you before that if you look at the olfactory behavior, we have these odors that the larvae don't like. And one of them is geranyl acetate that I already told you about. It's in lemongrass. And another odor we found that the larvae don't like is menthol, which is also in green leaves. It's in peppermint. And again, they don't like it, but if they're food deprived, they are starting to get attracted to it. But also here, we don't know, is actually peppermint something that's nutritious for them? Could they live on it? We don't know that. So we are wondering if we can look at the behavior that's maybe ecologically more relevant and actually provides them with food, but normally they wouldn't eat it. And there we thought about cannibalism. And there has been some publications about this phenomenon in larvae, that they do actually cannibalize on each other, on conspecifics. So you see here on the top right that they have these mouth hooks, and they can really, if they want to really bite into bigger larvae, for example, usually the small ones eat the bigger ones because the bigger ones at some point start slowing down before they pupate, and then they're an easy target. And so they do it. And so we were wondering if we can induce this behavior in our setup. So normally we have these traces. The larvae start out in the middle, and then they just roam around and explore the arena. So what we did now is we added 10 injured and dead larvae in the middle of the arena, together with our normally 15 alive larvae, to see if they would stay around and feed on these animals. And we really quickly found out that they're not super interested in these dead larvae, especially if they're intact. So we freeze them to kill them, and then we put them in the middle. And if we don't injure them, if they're intact, like the skin is intact, they really ignore them. But if we injure them, we see that some of the larvae actually stay and start feeding. This you can see here. So the red data shows that if you put 15 larvae, usually 10 to 20% are feeding on the injured and dead conspecifics. However, if we would put if we put normal fly food into the middle of the arena, then we see around 70-80% of larvae feeding and staying in the middle. So it's not just that they usually want to disperse and don't like to be in the middle of the arena, but it's rather that they really are not too much interested in eating conspecifics. And then we were wondering if they're used to the normal fly food. So could it be that having suddenly conspecifics as a food item, it's just novel and they're not used to it? So what about if we provide them with other insect larvae? So we choose some larvae that are about the same size, which are ant larvae, which we got from the Klein-Eidam lab. Our neighbors here, they work on leafcutter ants. And we also tried honeybee larvae, also from the neighbor lab, from the Nouvier lab. They work on honeybees. And what you see here is that they really loved it, the fly larvae. So they eat them like as they would eat the normal food. Even though, for example, the ant larvae, they get fed with this fungus, I think, from the leaf cutting ants. So they might also not be the best food of choice. But yeah, here they didn't hold back. There was no inhibition. They just started eating and really loved it. So we think it might really be something that they recognize that it's their conspecifics, at least it's their own species, and there is some inhibition that they don't want to eat that. And so this was interesting because it's something that they can eat, and it's also providing them with nutrients, probably a lot of protein, but they don't really like to do it when they're fed. So how is it when they're food deprived? And so this is what actually, so David did this experiment. He was doing an advanced course in the lab for like 3 months, and he did all this data. So it's amazing. So he looked actually, and he managed to food deprive these larvae for 3 days and still had some that were alive. That was very surprising. Anyway, but so what you can see here is that if you food deprive the larvae, they really get more interested in feeding on the dead conspecifics, and we see about 50% of the larvae suddenly start feeding. So a significant increase. And the same actually we saw if we provide them fructose. So again, we do this food component deprivation. They're deprived of proteins, but they have plenty of fructose. And then they also increase their feeding. So this seems to be really protein deprivation dependent behavior. They're really looking for the protein here. Yeah, and then again, it's kind of a similar phenotype as we had with the olfactory system. So there's an odor that they usually don't like, but if they're food deprived, they like it. Now we have a food that they usually don't like, but if they're food deprived, they start exploiting it more. And that's why we asked, okay, maybe serotonin plays a role here too. So we tested the serotonin mutant in this setup. And you can see that the serotonin mutants, they do in the fat state feed a little bit on that conspecific. Maybe it's a little bit reduced compared to the wild type. But if you actually now look in the food deprived state, again, we see the increase in the wild type larvae, but the serotonin mutant doesn't show this increase. So again, here also serotonin seems to be the neurotransmitter that's required for this food deprived effect. And so kind of as a little summary, I could show you that I think the Drosophila larvae, they are very flexible in their behavior. And they can very quickly adapt to new situations. So the refeeding, 10 minutes of feeding worked to reverse this behavioral switch in the olfactory behavior. And then also the food deprivation is really affecting a lot of behaviors. So not just the olfactory behavior, but also the actual locomotion of the animal, and also the feeding preferences, if you think about the cannibalism. And we see that some of these behavioral effects, if you look at the neural circuits, they seem to share the same neurotransmitter. But what I think is happening here, that it's probably different serotonergic cell types that are then required for the different, behavioral outputs, which I also think makes sense and would be kind of efficient, as you can then easily modulate the subsystems. So, for example, the refeeding then might just modulate the olfactory behavior, but they might still have increased locomotion. So this is all something we're now looking at, if we can find shared circuits or isolated circuits that are also set up in a hierarchical manner in this system. Okay, and I think that's it, and I'm also out of time, I guess. So this is my thank you slide. So I want to thank Whole Constance, the Center for Collective Behavior, people in my lab. So the lab just grew with 2 new PhD students, Hari and Akila. They literally just started. And I was lucky to be part of a research unit in Germany where we look at the modulation of olfactory behavior across phyla, which is also super interesting. And I want to thank you. And please go and check out the neurobiology groups at University of Constance. There's always some open positions. And as you see here, Constance is really beautiful, and it's a really, really nice place to stay. Yeah. Okay. Thanks a lot. That's it.
Thank you very much. That was really great. I have a few questions here. Are larval glomeruli populated with ORs of multiple types? That's came earlier in the talk.
So the larva is kind of special in that it's a really one-to-one system. So you have one olfactory receptor neuron usually expressing one olfactory receptor. There's, I think, 1 or 2 outliers, but there's like one that has 2 receptors, but usually it's 1 receptor. And then this one olfactory receptor neuron goes to one glomerulus and connects to one of the uniglomerular projection neurons. And so in these terms, it's special. It's a very, very reduced system. It's basically you cannot reduce it much more. It's just one ORN, one UPN, and that's it. And in the fly, you have, I think, at least like 60 ORNs that express the same receptor, and then all go to one glomerulus. So it's like going from 60 to 1, and then expands again in terms of projection neurons. But in the larva, it's really 1 to 1, and to 1, which, yeah.
Another question from Rory. Great talk. Do the neurons that you studied in the olfactory circuit from the first part of the talk retain similar roles in adult flies?
So that's a good question, because I think we don't really know. I mean, the uniglomerular projection neurons, they kind of still have the same job. I think their main job is to provide the higher brain with information for learning, and the mushroom body and information for like more innate behaviors, maybe in the lateral horn, even though it's still not that simple. And the uniglomerular projection neurons do the same in the larva. So I think they are doing the same, probably. For the local neurons, I think there's also similarities, because we have also these global inhibitory neurons in the antennal lobe, which we also find in the adult fly, which is basically they do the gain control. And this is, I think, the same task. Then we just start to understand all the other local inhibitory neurons, and I think we still don't know yet really what's going on here. Also in the fly, it's a bit different, that I think the olfactory neurons, they go to both antennal lobes, if I remember that right. And in the larva, it's like unilateral, so they stay on one side. So I think there is some differences in terms of that's the system in the fly is more adapted to flying, where you maybe need to compare more what's left and right of your antennae, and there's some wind information coming in. So there is clearly also differences, because the systems are adapted to, I think, the ecology of the animal, which is very different from larva to fly. And yeah, the CSD neuron, it's there in the adult too, but it has a little bit of different connectivity. So, and I think that's why it also has probably a different function, and is maybe also more required for sensitivity during flight for specific odorants. So yeah, I think that's pretty cool, because in some of the neurons, I think they just stay the same, they just grow. But then there are changes in connectivity patterns, clearly. And I think it also makes sense that the fly has different tasks to master than the larva. So yeah, like in the larva, we see a lot of this probably gustatory sensory input into the antennal lobe, which maybe is not that prominent in the fly. But it also makes sense for the larva, because it just sits in food all the time. So olfaction and gustatory systems, it's just all chemosensation, I could say. And it makes sense that it's all a little bit mixed. But I guess in the fly, this is also more separated, maybe. Yeah, that's how I think about it. Yeah. Okay.
We have a few more questions here from Gayatri. I hope I pronounced that correctly. There have been studies about starvation-induced nociceptive tolerance. Could there be any hierarchy regarding what happens first? Locomotion changes, cannibalism, and maybe nociception?
Yeah, probably. I mean, that's a good question. I guess if you're hungry, the first thing is to find food. So yeah, maybe it depends on how hungry you are. So maybe if you have different starvation times, the different behaviors are more prominent. But I guess in this, I mean, yeah, starvation-induced nociceptive tolerance makes sense, that you ignore a little bit about the temperature, because you want to explore and find food. And the hierarchy, yeah, I guess it's a bit hard to say, because, yeah, I was just thinking about an experiment that you could disentangle this, but might be difficult. I think, yeah, this just happens. Everything just happens. And maybe if you look at the neural circuits, we can find things that are modulated earlier on the sensory level. Maybe the nociceptive tolerance is modulated really on the sensor, that it's suppressed. And then you could maybe conclude from that, like where in the brain it happens, like what is more severe of a change. Yeah, that's what I would say there. Yeah.
I mean, I guess you could kind of monitor the nociceptive receptors and see if they are less active during this food deprivation. Yeah.
Exactly. Yeah.
Minel, I wonder what neurons project to CSD. I don't remember if you touched on this.
So it's a lot of neurons, I can tell you. So based on the connectome, there are some neurons we know, like for example, how we know how the olfactory information, like why it responds to odors. So it receives a little bit of input directly from like 2 olfactory receptor neurons. But it's not very broad. But what we see that it responds to a broad set of odorants actually. Like we tested a few, but it responded to almost all of them a little bit. And so what it also receives, it receives lateral horn input, which is like the higher processing center where also olfactory information is present because it's provided by the UPNs. And so actually what we think is happening in the food deprived state, that there is like this excitatory feedback loop that's closing. So the UPNs bring the information to the lateral horn activates the CSD activates the UPNs, and then you have this excitatory feedback loop that provides your enhancement of the UPN pathway. So this is, I think we know like how it responds to odorants. And then it receives some input from neurons in the antennal lobe, from these inhibitory neurons that I talked about, these picky local neurons, a little bit other local neurons. It receives a little bit of input from non-olfactory sensory neurons, maybe gustatory in the antennal lobe. And then it receives input from a lot of neurons in the higher brain around the lateral horn and the mushroom body region, which, however, we don't know much about. Yeah, we know that some are lateral horn, but then there's also some maybe indirect information coming from the mushroom body output. So it's actually a kind of a convergence neuron which collects information from lateral horn and mushroom body and then feeds it back to the early sensory processing. And yeah, I guess the question is also how the hunger information is kind of facilitating this response, but we don't know yet. So this is also something we're now looking into to really see what's going on there. Yeah, we don't know yet. Stay tuned. Yeah.
And Divya, finally. That was a beautiful talk. Looks like food deprivation, acute or chronic, is a complex manipulation. Is it possible in your setup to image multiple regions or whole brain to see changes in non-olfactory neurons?
Yeah, I guess you would see a lot of changes if you would do this. And yeah, we could do that. We could just genetically express GCAMP basically in all neurons in the brain. And I think people are doing this right now. So this is actually an approach people are doing. There was just a really nice paper from Daniel Mönch about the fly gustatory system, where they basically imaged just the whole gustatory system to see all the effects that happen in food deprivation and change in mating state, which is super, super exciting. And I think, yes, this is a good approach to get a general idea what's affected. I would guess everything's affected somehow. So I think we also need the more precise approach to really look at the single neurons and what's actually going on there. Yeah, but this is definitely, it's kind of where I started in my postdoc also with the approach. So where in the brain do we actually see a change, and can we pinpoint it to a specific system? So yeah, this is definitely a super, super good approach. Yeah, yeah.
We've run out of time. Yes. Thank you so much, Katrin. Thank you so much, the audience, for participating and for joining us. If anybody has any more questions, please join us on Zoom. And we'd be happy to see you guys next week. Thank you very much. We'll start in a few minutes on Zoom.
Okay, see you. Bye-bye.